Welcome to the MEF2C cast, the podcast advocating for the MEF2C haploinsufficiency community by broadcasting the voices within. I'm Eli.
And I'm Haley. We are parents to a boy with MCHS and autism, sharing our journey with you throughout conversations grounded in learning, support, and community.
This is Advocacy Amplified. Do you have questions, comments, or a story to share? Want to be a guest on the show? Connect with the MEF2Ccast on Facebook, mef2ccast.com, or email us at mef2cast@gmail.com.
If you enjoy the show, please support us by subscribing to the show on Apple and Spotify, leaving us a review, and following us on social media. You can also buy MEF2Ccast merch at our Shopify store, linked in the podcast description. The MEF2Ccast is for informational and entertainment purposes only. Views expressed are those of the hosts and guests and not necessarily of the podcast or its sponsors. This is not medical advice. Consult a healthcare professional for medical concerns.
Alrighty, hi everybody, and welcome to the MEF2Ccast. Our guest today is Dr. Laurence Hurst, Professor of Evolutionary Genetics at the Milner Center for Evolution at the University of Bath in UK. Dr. Hurst is an elected member of the European Molecular Biology Organization, an elected fellow of both the Academy of Medical Sciences and the Royal Society of London, and a winner of the Genetics Society Medal, as well as the Humboldt Prize. He is also the author of The Evolution of Imperfection, The Science of Why We Are and Can't Be Perfect, published by Princeton University Press, which was called Near Perfect by the Wall Street Journal. How are you this morning?
Very well, thank you. And it's a real privilege to talk to you and your community this morning.
Oh, I really appreciate that. The feeling is mutual. So can you start by just telling us sort of how you got into evolution and then, tell us about the sort of classic Darwinian model of evolution and sort of the questions that raised for you?
Absolutely. Yeah. So actually, when I was first taught evolution, I actually thought it was rather an obvious, dull subject. So, for example, we often teach, I do this to my first and second years as well, here when I'm teaching at university. We often teach one sort of classic case history. We've got plenty more now. But the simplest idea, and this classic story, is about black and white moths. They're the same species, but some are black and some are white. And what we know is that in the 19th century in northern England, where the trees went black because they got covered in soot, the black version became ever more and ever more common. And then come the 1950s, when in Britain we had this thing called the Clean Air Act was introduced, the trees started going pale again. And then alongside this, the moths increasingly became more and more pale. And it turns out the explanation for this was Darwinian natural selection. So I was basically told that if the trees are black, then black moths don't get eaten as much as white moths. So we'll leave more kids than the white moths because it's heritable, it's genetic. That means that over time, over many generations, the black goes up and up and up, simply because they're dying less and they're leaving more kids than the white versions. So then come the 90s, the process goes into reverse because the trees go white. So now the white ones aren't seen against a white background by the birds who eat them. That's the major predation here. And so the white ones go up in frequency because they're leaving more kids, and those kids will leave more kids because they're more likely to survive, and so on and so forth. So there is sort of the canonical view of evolution in many regards. It's the sometimes called survival of the fittest, the ones who are well adapted to the environment, the ones who fit into the environment, so white against the white background. The ones who fit into the environment are the ones who we're going to expect to see more of in the next generation. It's all about changes of frequencies of mutations. So there's a mutation that makes you white, another that makes you black. And what is the relative frequency of those two in the population? Now, if you take that, and that is our canonical view of how evolution proceeds. And it's a very nice, and there's, in a sense, nothing wrong with it, except it's incomplete. It's incompleteness that is the problem. And where it becomes particularly problematic is if you try and think, okay, what's the logical corollary of all this? So if that is the only sort of process going on, and it's just survival of the fittest, you would, I think, expect that things should evolve towards perfection. So if the trees now do not change color at all, we should expect, and the trees are white, we should have ever better camouflage and ever better camouflage until the point where you just, nothing can see the moth against the background. It's so brilliantly camouflaged. And when you look at camouflage, much of it is absolutely fantastic camouflage. You go, hey, that sort of makes sense. And we sort of have a metaphor for all of this. I don't know if you put the word evolution into Google image search, this is quite a fun thing to do. nearly everything that comes back is that image of, the chimp knuckle warming, walking, then increasingly becoming upright until you get to man, going left to right. It's called the march of progress. And the idea is that it's a progressive process. But also, not only that, one of the biases in that image is that humans are sort of the top of the evolutionary Christmas tree, as it were. And it's all sort of speaking to this same underlying notion that evolution is a progressive process, that the end of evolution is somehow a state of perfection. And so what really started to intrigue me was, particularly with humans, and I think this is really the focus of the book in many regards, particularly for humans and human genetics. When you look at our genetics, you go, that's not obviously perfect. Let me give you a couple of examples here, for example. So, with the ability to really get at genetics now, we know, for example, from women who have cancer and have to have their eggs frozen to be fertile, for example, you can go and look in those eggs if they're not used, or the neighboring polar body, which is never used, and you can count up the chromosomes. And it turns out that one study which did that found that 70% of eggs that have been released by a woman, 70% have the wrong number of chromosomes and will die instantaneously. And when we look at the early human embryos, we're getting similar numbers. On the average, it looks at about 50%, half of all human embryos. So fertilized egg makes a zygote, so the first cell, most of those will die very, very rapidly because they've got the wrong number of chromosomes. And you go, that's not obviously the result of a perfecting process. And likewise, in fact, pregnancy, generally speaking, in humans is so far from perfect. It's quite remarkable. So we have extraordinarily high miscarriage rates, for example. So even when an embryo is recognized and you have a recognized pregnancy, so post week 6, we have about 15 to 20%, rising to 67%, 70% in older women, but about a usual 15 to 20% spontaneous miscarriage rate. We have diseases of pregnancy like preeclampsia that affect 5% of pregnancies and potentially kill mum and offspring, and that's a human-specific disorder. But then when you look at our DNA as well, it just doesn't obviously speak of perfection. One simple example, so your audience, you know about genes. So what do genes do? Genes make RNA, the RNA of RNA vaccines. Genes make RNA, and RNA gets turned into protein, and it's the proteins that are the sort of doing molecules of the cell, you might say. And you go into a bacterial genome, and so all the DNA of the bacteria, and you go, okay, how much of that is doing that? How much of that is gene, makes RNA, makes protein? And that's about 85% in, if you take the E. coli that sits in your gut, a lab bacteria, it's about 85% of their genome is DNA that makes RNA, makes protein, and the other 15% or so are the control elements for the most part. Things that, the switches that sit right in front of a gene, so they turn on, turn off. So most of it seems to be functional. Now, you do exactly the same for the human genome, and our best estimate at the moment is that the amount of DNA in our genome that codes for protein is about 1.2%. So not 85%, 1.2%. At which point, there's a really interesting problem there. And more generally, if you ask about genome bloating, how much other stuff is there in the genome, we've got a series of independent measures of that, and all of those independent measures co-vary. So, for example, our genes have lots of introns in them, which have to be snipped out of the RNA to make the protein. The size of ours correlates with the size of intergene DNA. So our DNA not only has small protein coding bits, the bits within the genes that aren't protein coding are big, and the bits that sit between genes is also big. So if you sort of plot out how big are those two things across species, those two things are independent, what you see is that across species the two covary. So something like our lovely laboratory yeast, for example, has very few and very tidy little introns and very small intergene distance. It's a compact live genome. If you can think of a beautifully engineered Swiss watch as a genome, that's the yeast genome. It's glorious. It's very nice, tidy, compact, well behaved. Ours is just bloated, and it's full of other stuff that's not obviously doing very much. So I said 1.2% of our DNA is protein coding. 8% is the remnants of old retroviruses. Things like HIV can insert into our DNA, for example, and 8% of our DNA is old retroviruses. They're called endogenous retroviruses. They're dead, and nearly all of them are dead, but they just sit there, and another 40-50% is so-called transposable elements, jumping genes. I think they're quite closely related. So it looks mostly like a graveyard for old infections and jumping genes and stuff we don't really need.
So does all that like, is there a way that organisms get rid of that DNA that they don't need and we're just not doing it, or does that just accumulate over time as things?
So that's a really great. This is the profound question. Why
Mhm.
is it that our DNA is full up with stuff that it looks as though we don't need? Occasionally, some of this, I actually work on some of this. Occasionally, it looks as though some of it gets reworked for us, for example.
Mm.
But most of it isn't. Most of it appears to be dead and decaying rubbish. So there's the question: why is our genome full of dead and decaying rubbish? But the yeast genome is not full of dead and decaying rubbish. What's the difference? Is it because they don't have transposable elements, retroviruses? No, they do. So we think that the real key process is actually an evolutionary process, not it isn't happening, things are inserting in. So the evolutionary process we're thinking about here, think about it this way. So transposable elements, a big block of DNA or a virus or something inserts into your DNA. That's happened in one individual. Yes, it's not a species-wide event. It's happened in one individual, and it's happened in the DNA that makes sperm or egg. So it is potentially transmissible through to the next generation. So it's made your DNA a little bit bigger. It's made your DNA a little bit more expensive to copy, but it's probably not a disaster. So it's probably what we call weakly deleterious. It's not, very occasionally, they will cause a genetic disease. If one of these things inserts into a really important gene, that can cause a genetic disease. Most of the time, they just go and insert in barren wasteland of our DNA. So, yes, it's made it a little bit more expensive to copy our DNA. And it's made it a little bit more expensive for yeast to copy the DNA. Now, why would those two things be different? Well, at this point, I need to introduce you to one of the really key concepts that is changing our view of the evolutionary process. So I gave you the canonical view of evolution. That good things happen in populations, good mutations happen, they spread. Bad mutations happen, they get kicked out of the population. So if, for example, imagine all the trees are white, every moth is white, and in comes a black version, what's going to happen to the black version? Well, in the canonical view, it's harmful. Yes, you're going to get more likely to be eaten. There's no way you're going to take over that population. Over time, the black variant will simply be kicked out of the population. That we call so-called purifying selection.
Right, yeah. So sorry, just to interrupt you for a second. So just to back up a little bit. So mutations occur randomly, and
Yep.
then natural selection is the process by which these mutations are chosen for
Or
to
not.
increase or not to increase the fitness of
Yeah,
any
so
organism
mutations will have
on.
effects on fitness, is the
Right.
bottom line.
Yes.
And the
And
canonical view is if it increases fitness, you go up in frequency. If you decrease fitness, you go down in frequency. And I think conceptually that's easy to see. So
right.
the mutation that makes the moth black when the trees are white, that will go down in frequency because it's more likely to get eaten.
Right. And just one quick point I want to make. Actually, I just have an undergraduate degree, but it's in conservation biology, so I know a little bit about this. And one of the things that a pet peeve of mine is when people assume that like the moth is like, oh, the trees are darkening, so I'm going to turn dark,
Yeah,
and
no,
that
no,
will
no.
hide me from the birds. There's no conscious effort in these things. It's just, it's
No, we're mutating all the time. Everything is
right.
mutating all the time. There's no evidence that any mutation is directed in the sense that I've got a problem today. So you can imagine we switch, and you can do this experiment in the lab, it's been done plenty of time. Grow bacteria, for example, grow those E. coli in your gut up on glucose, and then suddenly switch them to fructose. Now they've got a problem, and the problem is how am I going to eat fructose because it's a different
Mm,
sugar? And it turns out all the time they were mutating all the genes and occasionally making something that could metabolize fructose, but it's no use when you're in a glucose-rich environment. When you switch them to a fructose-rich environment, those mutations are still happening all the time. But now the same mutation that affects fructose, now that's useful. It's suddenly become helpful. But you don't make it simply because you're in a new environment where you need fructose. You don't become black. You don't have a black mutation or a white mutation, whatever. You don't have that because of the new environment. They're happening all the time. And this, as I said, we can do this very nicely in E. coli, in bacteria, because we can do it in real time in the lab. So you can see all the time in the lab, the mutations just going, pinging off all over the place. But most of them get eliminated. And over time, what you expect is the ones that are good go increasing in frequencies. Over time, the E. coli will just get better and better and better at living under lab conditions. This has actually been done by a bunch in America in a long-term evolution experiment. They just grow E. coli up in parallel, lots of different lines, and over many, 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 many generations. And they see indeed it slowly adapts to the new environment of living in a test tube, as it were, and gets to a point where it's not really improving very much, but it improves rapidly to start with. So yeah, these mutations are happening all the time, and in these sort of perfectly adapted E. coli, or any perfectly adapted species, you're still mutating. It's happening all the time. You, in fact, were born with somewhere between 10 and 100 mutations, new mutations somewhere in your DNA. So yes, mutations are happening all the time. They're not directed, but what happens is selection is then acting as a filter. And if it's an advantageous mutation for your environment, the expectation it will increase in frequency, the white moss when the backgrounds are white. If it's a harmful mutation in the current context, it should go down in frequency, and so on and so forth. And that's sort of canonical model. Mutation is just happening all the time. It's a sort of background process. Selection is a filter. So you could think of it just as well as, you're randomly making beads of different sizes all the time, but the filter then is a sort of sieve, and some things will pass through the sieve and some things won't. But you keep on making balls of different sizes. Some things will pass through, some things won't. So in the canonical view, the really simple, nice canonical view, to increase in frequency, you have to be good for the individual. To go down in frequency, you're bad for the individual, and that seems very sensible. What's turned up in the last 20, 30 years or so is a body of theory which says actually there's more to it than that. Because what you're looking at there are sort of average effects. On the average, that may well be true. What's been pointed out is that if these mutations are of small effects. So notice we're here talking about small effect mutations. So let's suppose this transposable element and it inserted and it reduced your fitness by, I don't know, 1 in 100 million. So you're pretty, you won't notice any difference. There's no way you're going to turn up to the doctor because of this. This is just a very minor effect mutation. What's going to be the fate of those? Now, what was pointed out by a Japanese population geneticist, very nice lady, Professor Ota, what she pointed out is actually the rules of evolution can now change a little bit. And they can change a little bit because when things are very, very small effects, those deterministic trends may not now apply. And the best metaphor to think about this is to think about the deterministic ones. Imagine we're thinking about particles of dust. So when the light comes in through a window and you can suddenly see the dust in your room, and those dust particles are moving up and down. Yes, they may be a little bit heavy, but they can still move up, even though on the average they should move down because they're a little bit heavy. So that's the analog of moving up because you're advantageous, moving down because you're deleterious. But here's something that's actually deleterious, meaning heavy in this case, but still they're moving up. And actually what she does is she borrows the maths of so-called diffusion processes like this. And what she shows is a really important result for modern evolutionary theory, which is that actually the ability of selection to get rid of harmful things is much reduced if the population is small. And intuitively, perhaps you can see that because small populations are going to be much more vulnerable to chance events. So this is like asking the question, imagine you have a dust particle and it's just a little bit dense. So on the average, it should go to the floor, not the ceiling. But what's the chance that it is actually going to get to the ceiling? Against the direction of gravity, against the direction of selection in this case. And of course, I think you can see if the room is it's not tall, it's quite short, actually just a couple of little bubbles about, and you could actually have something that's not good for you, not desperately bad for you, just a little bit bad for you, but it could still hit the ceiling. Whereas in a very large room, that's not going to be true. And the analog is, so small species, we think this selection to get rid of harmful things is actually a weaker process. It's a less efficient process than exactly the same, but in a much larger species.
Do you mean
Larger meaning not
small?
body size, by the way. Larger
Okay,
larger simply meaning how many individuals are in the species. As it happens, those two things almost always co-vary because large species have small population size. You don't get massive populations of whales and elephants, but it's the population size that matters. And so we've done a whole series of tests of this sort of thing. So does the population size predict, for example, genome bloating? So the
Mm,
idea would be, let's go back a step. You have this new transposable element. In one case, so it's only in one individual in humans, and it's only in one individual in yeast. Is the fate of this transposable element, this extra bit of DNA, the same? So it's going to reduce your fitness in both cases, let's say by 1 in 10 million. Minuscule effect. But because yeast's population size is so massive, they're tiny little things, there's billions of things. According to Ota's calculations, that means that actually that is simply going to be effectively deleterious in the sense that there's no way that's ever going to get through to fixation. That's a massive population size. But for us, it is. Our population size is estimated, the ancestral population size, that is, when all of this was happening, not so distant past, hundreds of thousands of years ago. Our population size is probably a little bit under 10,000. I mean, it's always been, but there's billions of us. Yes, there are now, but before
mhm.
there weren't. Um, so it's about 10,000. And anything having a selective effect, which just reduces your fitness by 1 in a million, that is just now basically a dust particle that can float up or down. Whereas in yeast, exactly the same thing can only really go down. And so in us, it can go from rare to common, not because it is good for us. And that's the key point. It's not because it is good for us. It is simply because selection is an inefficient process
Mm,
against these weak effect things when population sizes are small. And as I said, what we find is that if you look across species, the size of those introns, the size of the intergene distance, the amount of genome bloating, for example, that's all really nicely predicted by the effective population size. So our small population sizes, much more easy for dust particles to go from the floor to the ceiling because it's a short distance from floor to ceiling. Not many lucky jumps for my rare new mutation to happen to jump up and get to a high frequency. In a large population, it's not going to happen. There too many lucky jumps are needed, as it were.
yeah. The and this may be more telling about me than anything, but the example that came to mind when I first heard about this is I grew up, we had a bunch of property and a great big yard, and we had this like 1970s wheel horse riding mower. And to start it, you would have to jump the solenoid. So we had a homemade jumper cable, and anytime it stalled out or you ran out of gas or whatever, you'd have to jump off, pull this jumper cable out from under the seat, and jump the solenoid, and it'd start up, and then you could get going. Now, if you were an alien anthropologist who had never seen a lawnmower before, you might assume, well, that's the best way to start the lawnmower. Well, it's not. It's just not such a pain in the butt that it's worth us to go get it fixed. You know what I mean? So it is
It's a very,
deleterious
very
in the
nice
sense of the
metaphor,
gene,
actually.
kind of like the gene, but it's just not so deleterious that
Yeah.
um
So we actually we refer to it as quotes junk DNA, and the idea junk is not the same as trash. So if you imagine that you have to pay something to get certain things taken away. Yeah. So, here's a block of wood. I could put it in my garage. I could put it in my loft, or I could pay for somebody to take it away. What am I going to do? I'm going to put that in the garage. It's
Yep.
costing me nothing. It's taking up a little
Mhm.
bit of space, but I'm not going to pay somebody to take that away. That's a complete waste of money, as it were. But yesterday's rotten dinner, yes, I'm going to pay
Yeah.
somebody to take that away. And so that would be the equivalent. The rotten dinner is the equivalent of the highly deleterious mutation. Yeah, selection is going to get rid of that, and yes, I would pay for it to be taken away, as it were. And in a sense, what it says is, the calculation for yeast and the calculation for us is different. That the yeast will get much closer to the perfect lawnmower because actually the selection is so good it would get rid of the crap lawnmowers and it would only favour the really good ones. Whereas with us, we just tolerate not very good ones.
Right. Yeah. Yeah. Junk is not the same as trash. And
Junk is not the
I
same as
love
trash.
that. Yeah, so it seems like maybe, at least according to this theory, that like again, if you think of selection as a filter and there's like a spectrum with like the most advantageous alleles being on the right and the least being on the left, what you would expect with the canonical model is that that filter would be right in the middle,
Yep.
but maybe it's skewed more to the left
Yeah,
than you would
so
expect, at
so
least
exactly that is
according to
exactly it.
Otis'
So
theory.
if you take the really simple canonical model, you only increase in frequency if you're advantageous, and you always decrease in frequency if you're deleterious. What this is saying is, as you get closer to that cutting point,
Mhm.
then what is happening is it's not selection going yes, no. It's these chance processes going, oh, you can still increase in frequency because you're harmful, but you're not that harmful. And so, it's the difference between a particle of dust, which I've attached a lead weight to, boom, straight to the floor. There's my deleterious thing.
Mhm.
Versus a particle dust, it's a little bit dense, but we're now close to it's almost zero effect. But because it's just a little bit dense, it can still bobble about and go up in frequency. And if you're bobbling about and you're going up in frequency, this is mutations going up and down in frequency, just to reiterate, so becoming more common, even though the canonical model said, oh, you're deleterious, you should be going less common. But actually, no, there's a chance that you can be more common. And then the chance that you go from very rare initially, because all mutations start out rare initially, to actually everyone in the population has it, that can actually be quite high if the population size is small, and it's very, very low if the population size is really big. And that is really actually where evolutionary theory is at the moment to understand genomes. That's our major model, because it seems to be getting just about everything right. So it says that actually when you're looking at a species like us genetically, we are not perfect, and we shouldn't. We would not be expected to be perfect. We should be the ones who are accumulating these mild, of really tiny effect transposable elements. They're not doing any great harm, but our selection just simply isn't efficient. It's not that they will always increase in frequency. It's just that the test has been done all the time. Transposable elements. We almost certainly all were born with a couple of new transposable elements somewhere in our DNA, for example. And so that test to be done all the time, and some will increase by chance, some will decrease by chance. But the dominant process is not this: you're increasing because you're beneficial, you're decreasing in frequency because you're deleterious. Actually, the dominant process is you're just randomly moving up and down because selection is inefficient. It can't push you out of the
Mm.
population because it's too weak a process.
One of the other things that I thought was really interesting is that humans have a much higher mutation rate than you would expect.
Absolutely. Well, it's a really great point, because one of the corollaries of this, so I said one of our predictions would be that, for example, we've got a small population size. According to Otis, selection should therefore be efficient. By the way, this is called the nearly neutral model of evolution. It's by far, she won. There isn't a Nobel Prize in evolution, more's the pity. But there's a thing called the Crawford Prize, which the Swedes put instead. And Ota won the Crawford Prize for this work. It's by far the most important contribution to evolutionary theory in the last 20 or 30 years or so, because it says actually things that are harmful can still spread, but it all depends on the population size. So everything we look at then seems to be varying with this population size effect, so the amount of genome bloating, but also when we process our genes, we have to cut them up, splice them. The number of different splice forms, because we're getting that wrong all the time, that's going with the effective population size. So whenever we do our gene processing, we're getting it wrong a lot of the time, not because there's some great advantage in getting it wrong all the time, just simply because we're rubbish. Selection isn't strong enough to go, this is how you properly process this gene every time. Now, yeast, they always get it right. They never seem to make a mistake. And so again, it's not just a genomic anatomy that we're concerned about. How the genomes behave, how they processes genes as well, seems to be very nicely predicted by this. But as you say, the really interesting one, I think, is the extension of this idea, which says that actually now consider something else. Consider a mutation that slightly increases your mutation rate. How's that new mutation going to behave? And what this theory says, actually, if you've got a small population size, well, actually, that's going to be a weak effect. It's changing your mutation rate, but it hasn't changed it a lot. So you're going to that has a chance of slightly increasing in frequency. Whereas in our beloved yeast or in bacteria, for example, their population sizes are so huge that if you had a mutation that increases the mutation rate, they'd almost certainly be heading down in frequency, because all they've done is made more deleterious mutations, and selection is really efficient at getting rid of that. So the same body of theory predicts variation in the mutation rate. Now let's be clear what we mean by mutation rate here as well. So in this case, what we're talking about is the per base per generation. So we got about 3.3 billion bases. Now, of course, you'd expect us to have a higher mutation rate in the genome as a whole and all the DNA, simply because we've got a lot of it. So that's not what we're measuring. That's the mutation rate per base pair times the number of base pairs, as it were. So to get rid of that variable, how many base pairs you have, we calculate in terms of per base pair. So if you have an A, C, T, or G in us, what is the chance that your kid is going to have, so let's say mum is A, dad was T at this particular site, what's the chance the kid is something else? A mutation either in mum or in dad, and the kid is something different. at this particular residue, for example. And what this theory then says is that just as we expect genome bloating, genome inefficiency when populations of sizes are low, so too we expect higher mutation rates because selection is less good at getting rid of mutations that increase the mutation rate, because those also are going to be rather weak effect mutations. And so one of the things that's turned out in literally, I guess, the last few years, because now we have the ability to do something absolutely amazing, which is you can, for lots of species, you can sequence mum, dad, and kids. You sort of do a spot the difference competition, and i.e., where are the kids different from mum or dad in the DNA that they inherited from mum or dad. And from that, you can say, okay, they've got a certain number of new mutations, divide that by the total genome size, and you get a per base pair, per generation mutation rate. Theory says we should have a high one because we've got a small population size because we're big organisms. And it says yeast should have a low one because they've got a high population size, so they're very efficient. So we can now do this for lots and lots of species. And when we do this for lots and lots of species, and you ask what actually is predicting the variation. So some organisms have mutation rates down at 10 to the minus 11 per, mutations per generation per base pair. Your average bacteria, so E. coli, is about 10 to the minus 10. These are very, very low numbers, by the way. A fruit fly is of the order of 10 to the minus 9. Most insects are about 10 to the minus 9. And you ask, who's actually got the highest? Well, there's one strange fungus which uses mutation to kill transposable elements. We won't go there. Leave that one out of the equation. And the current record holder for the highest mutation rate of any species is, you guessed it, it's us. We come in at 2 times 10 to the minus 8. So 10 to the minus 6 is 1 in a million, minus 7 is 1 in 10 million. So it's about 2 base pairs in 100 million base pairs. If I were to take 100 million base pairs of DNA in a kid and go, what's the chance it's got a mutation in it? You'll get about 2 mutations in 100 million base pairs. Now, it's telling us 2 things. One is the mutation rate for everything is extraordinarily low. This is quite a miracle that it's that low. And we know why it's that low, because most mutations are harmful to some extent. So you increase the mutation rate, you're just going to cause harm. So we think selection almost uniformly says keep the mutation rate as low as you can, because most mutations are harmful. Yes, occasionally you will need advantageous ones to adapt, but that's not why you have the mutation rate as it is. Most mutations are simply deleterious. They may produce the occasional adaptive one, but most of them are deleterious, so keep the mutation rate down. That's what the theory says, and it's what we find. But that variation that we find between organisms is very nicely explained by effective population size, and so this nearly neutral theory, and ours happens to be the highest. And that equates to, as I said, we're all born with somewhere between probably about 10 and 100 new mutations somewhere in our DNA, which is quite a bit. So that's on the DNA as a whole from the 10 to the minus 8 figure. So that's quite high. So if you ask about the total number of new mutations that any genome has, now that will, of course, scale with genome size. And something like rice is probably competing with us. Rice has got a much bigger genome. The rice plant, that is, the rice that you eat, got quite a big genome. Wheat's got quite a big genome as well. So they probably have more per genome, but actually our rate per base pair is still the highest yet seen, even a slightly bit higher than whales. Strange to say, the whale number came out quite recently. But yeah, they were about 1 times 10 to the minus 8, we're 2 times 10 to, very, very close to ours. But yeah, so far we're the highest, which of course has knock-on consequences.
Is that, would you expect our mutation rate to be the highest based on like the predictive modeling that
We think there are 2 they're really good questions. So the whales problem is a particularly interesting one. If it was just population size alone is the important predictor, we know it's the big predictor. So if you work out what the effective population size is, work out the mutation is, it's a beautiful, beautiful match. But there's still variation, and the whales are just below. What we think is actually going on is that it's not simply weak selection, but the selection is going to be slightly stronger in whales, because if you inherit a few too many mutations and you're as long-lived as a whale, you're more likely to get cancer. So there's this second effect of, well, how long are you actually going to be living? So each new mutation that you have increases your chance of getting cancer, but your chance of getting cancer is also dependent upon how old you are going to be living to. Now, we don't live as long as whales do. Whereas mice, for example, mice will all get cancer by age 2 or so. So you can't keep mice living long at all. simply die of cancer. And that will also be a contributor to the overall mutation rate. So we think it's actually two things. One is the population size, and the other is actually how long you're then going to live. So whales could probably not sustain our slightly higher mutation rate because with something like the two-hit model of cancer, for example, the chance that you're now going to get the second hit is a little bit higher in whales because they live longer and so more opportunity for the second hit, whereas we don't live as long. So ancestrally, we probably live 40, 50 years. And if that's all that we're living, we could have one mutation, and the chances are we're not going to get the second. Whereas if a whale is living 150 years with the same mutation rate, the chance it's going to get a second one, which will kill it early for a whale, not early for a human, is probably that little bit higher. So we think the dominant effect is population size, and there's a second effect, which is how long you live, effectively protecting you from late-stage cancer, as it were. And the reason we get late-stage cancer now is our mutation rate is almost certainly not adapted to our current longevity.
Right. Yeah.
We are living way past. So the diseases that kill us are the first world diseases for the most part. So cancer, type 2 diabetes, heart conditions, and so on and so forth. These are things that in the past didn't kill us. So there's a very interesting case history is actually what did kill us. And so if you go to some of the Pacific Islands, for example, there's some fantastically interesting studies where they happen to be studying the Pacific Islanders and they die really quite early, 30, 40, 50, and it would typically be an infectious disease of some variety or another. What's particularly interesting about the Pacific Islanders is at one point then a lot of investment comes in from outside because of mining possibilities and so on and so forth. And so what was a poor rural population now suddenly becomes really quite a wealthy population. And so what happens with wealth is they now live longer, but they die of different things. So they're no longer dying of infectious diseases. They die of type 2 diabetes, of coronary heart disease and cancer. So you've got this lovely transition from you didn't have much money to you do have much money. And what kills you then is different and your longevity is different. So yeah, we almost certainly are sort of evolutionarily mismatched, as it were. We're doing too well, and so we're dying of first world diseases now. And so our mutation rate probably almost certainly isn't adapted to our late onset cancer probabilities because we never lived long enough to get cancer. Is
Right.
probably
Yeah, that totally makes sense. So that was a good segue into kind of tying how all this goes into your
absolutely.
genetic disease, but I'm going to trample on a little bit because I have just one question before that. So, is there any correlation between like ecological role and mutation rate? Because like with my very basic understanding of evolution, like it would make sense to me that like humans, for example, who were incredible generalists, I mean, we're on 7 of the 8 continents and thriving, whereas opposed to something that's more specialized, like if you were a generalist, it may be more advantageous to have a higher mutation rate because that would allow you to more quickly you're essentially throwing more volume. You're throwing more mutations at whatever environment you find yourself in, and you might be able to adapt to it. Whereas, like, say, I don't know, one of the Galapagos finches or something that's like very highly adapted to that environment, it would make more sense to me that the mutation rate would be
Yeah,
low.
so to a large extent, you're thinking the wrong way.
Okay.
What you're doing is you're thinking, why would it be useful to have more mutations? The core problem, you need to sort of stop thinking that mutations are there to enable adaptation. The core problem is that while it may be true that if you had a higher mutation rate, you may be able to find adaptive mutations more easily, you will also be finding deleterious mutations more easily. And that's why we think selection almost uniformly favors a low mutation rate. Now, there are one or two very exceptional circumstances. So you can do an experiment with bacteria, for example, where you make them co-evolve with just one bacteriophage in the lab over time. And in some of those cases, you can see evidence that there's been selection for an increased mutation rate to enable adaptation to resist the virus. But that's a very unusual setup. But for the most part, organisms do not adjust their mutation rate to adapt. Things that need to adapt do not have a higher mutation rate. And the reason for this is because you increase the mutation rate and all you're doing is killing yourself because most mutations are harmful most of the time. So you have to think, I think, much better way to think about mutation is that these are simply errors in DNA processing. You want to keep those at that error rate as low as possible. I mean, think about it this way. If I told you I'm going to allow you to go into your car's engine and randomly whack something in the hope that, you need your car to be a better car,
Mm,
you're going to go, don't be so stupid. I know I'm going to break my car.
Right.
Yes, you take something that's fairly well functioning, that says we are fairly well functioning, we're here talking to each other. You randomly change it, which is what mutation does. Most of the time, you will make it better. Now, I grant you, very, very occasionally, you'll go into your engine and you'll give a good old whack and it'll do something beneficial. But 9,999 times out of however many, you're going to make your engine worse and you're going to regret the fact that you went in and randomly tweaked it. Likewise, if you ever have a beautiful watch or something like that, I've got a lovely grandfather clock, but I'm never think of even trying to adjust
Mhm.
it myself because every time I do, I break the damn thing.
Right, yeah, no, that
Random tweaks to good functioning things will only make them worse. So you shouldn't be tweaking if you don't have to. But mutation is an error.
Mm,
Something is tweaking your engine all the time. Something is tweaking those watching all the time. Simply because of the process of making babies, you will make errors
right.
in doing that. It turns out it's exactly the same as if you had one of these old USB sticks that people don't use anymore and you drag a file from your computer to a USB stick, you're copying the data. Every
Mhm,
time you do that, you will be degrading the file. There will be bits of
mm,
information that get lost. Is that advantageous? No, of course not. You don't want that to be happening. You have to think about mutation more or less in exactly the same way. Most of the time, what it's doing, it's just random and it's breaking stuff. And that's why these rates are incredibly low. I mean, 10 to the minus 11, I think actually exceeds the error control capacity of these USB sticks. I think there was an old colleague of mine, he died a few years ago, one of the great evolutionary geneticists of the 20th century, a guy called John Maynard Smith. And he was asked on a program once to nominate what he thought were the great wonders of the world. And people were expecting, the Hanging Towers of Babylon or something, Gardens of Babylon. And he came up with a mutation rate. And his argument was it is so low. It is amazing that it is that low. And it's that low because most of mutations, most of the time, are harmful. So we have in our DNA lots and lots of repair enzymes to try and stop this happening. So the errors keep on happening, the repair enzymes keep trying to patch in, and occasionally things get through the repair enzymes and they end up as mutations. So don't think, I need to adapt, therefore I'll find a mutation. You find an adaptive mutation, you've probably found a couple of 100,000 deleterious mutations at the same time. So we know adaptive mutations are rare. The harmful ones are very common for very obvious reasons because these are well-functioning machines.
yeah, that makes complete sense, and now that I think about it, like, when a lot of times when I'm talking to other parents, I make the point that, women make hundreds of thousands of gametes and men make, for practical purposes, infinite. I mean, it's not actually infinite, but,
It's
mathematic
remarkably high. It's very, very high.
it's yeah. Um,
You need about 3 million sperm per mil per ejaculate to be fertile.
Yeah. So like those when you consider that and when you consider that we have what is it 3.2 billion base pairs,
Yep,
like
something like that.
it's actually really incredible that there isn't
A higher
more
rate.
rare genetic disease because
Yep,
there's just so many opportunities for things
to go
to
wrong.
go awry.
Yep.
But so kind of talk about how all this evolutionary theory you've talked about kind of ties itself together and factors into rare genetic diseases in humans, and then also talk a little bit about I heard you say that medicine is anti-selection, and I think that's a really interesting point. So talk about that
Yeah.
point a little bit.
So we have this high mutation rate. It's largely errors, nobody's fault. Selection can't do anything about it because we're a small population size. And so we are just mutating. Every one of us has 10 to 100 new mutations, not because they're good for us, just simply because you can't stop it happening. Now, what's the consequence of this? Well, what's going to happen to these mutations? Well, what we know is that some of these are going to cause disease. Some of them are going to cause genetic disease. And you can then ask the question, so these aren't mutations that are anything to do with a nearly neutral mutation. These are just simply harmful mutations. So what's going to
Mm,
happen to them? And then we meet another concept, and this is the concept mutation selection equilibrium. So there's an idea that mutation is forever pushing these things into populations, not because they're good for you, because you can't stop mutation. So at some rate, every genetic disease has new representatives every generation, and selection is, in a sense, knocking them back out again. So every time you have a tragic kid with an early cancer, for example, that you couldn't save, there's a mutation. The kid's died. That mutation is, in fact, not in the population anymore. So there's selection. That is negative selection. Killing a kid, the death of a kid with a harmful mutation like that is, in population genetic language, purifying selection, otherwise known as a tragedy.
Yes.
One language for population geneticists and fruit flies, and it's a personal tragedy for those who are affected.
Yeah, purifying selection
It's
is
a
a
horrible
really
word. It's a
yeah,
horrible word.
need to come up with a
Horrible
better
term.
term for that
Yes,
because
please, please. No, I always think I have to apologize for this language.
mhm,
It actually came, I think a lot of it did actually come from a rather unfortunate time when the eugenicists were actually thinking about purifying populations and
yes,
so on and so forth. And it's horrible language. It's
mhm,
fine for fruit flies. It is not all right for humans, I don't think. No, this is a personal tragedy when we're talking about humans, but the process in a sense is the same. It is selection. It's
mm,
selection against harmful variants. And it depends if the mutation is dominant or recessive. So the recessive ones can actually sit around at slightly higher frequencies because a lot of them don't get seen for quite a while. And then like cystic fibrosis, and the mutation comes out when you get two copies of it. And a good number are dominant, in which case, quite often with the dominant ones, it's the actual spontaneous mutation that generated it probably was either it happened in mum or dad when they were making sperm and eggs. Neither mum nor dad need be affected. They may well have generated it just by chance, not because it's a good thing, in their germline making sperm or eggs. And because we have a high mutation rate, we end up having a high number of these so-called rare genetic disorders. So you say, well, what sort of frequencies should you expect these things at? And the answer is very, very low. They should not be common in populations at all. These aren't mutations that are going to be increasing in frequency either because of these random processes or because of selection. They're simply sitting there because mutation pushes them into the population, selection pushes them out of the population. But there's a dynamic balance between those two processes. And in fruit flies, we understand it. And in humans, it's an absolute tragedy. But that's what's going on. What we do is we say, okay, well, let's classify a genetic disease as a quote rare disease. And the definition of that depends slightly on it's slightly arbitrary, but it depends slightly where you are in the world. The Koreans have a particularly low threshold, but both the EU and the Americans sort of largely agree. It's about a rate of about less than 1 in 2,000 incidents at birth, and you'd call it a rare genetic disease. And the oddity in humans is that when you make a compendium of rare genetic diseases, depending on how you count a rare genetic disease, there's somewhere between a count of about 7,000 up to 40,000. The variation depends in some part as to exactly how you classify something as being the same disease. So, for example, if you think about haemophilias, these so you cut yourself, you don't stop bleeding. You could say, well, that's one disease. They're all genetic, so that's one disease. Or you could say, well, actually, there's one flavor which is due to mutation in this gene. There's another flavor which is due to mutation in that gene. There's yet another which is due to that gene, and we'll label each one of those as a separate rare genetic disease. So depending on how we do the classification, there's somewhere between about 7,000, 40,000 rare genetic diseases that have so far been described. And those are only the ones that are being described. So there are probably others out there waiting to be discovered. And you then ask another question. I think this is the striking thing is you ask what proportion of the human population has a rare genetic disease? And you think, well, they're rare, can't be many. And no, because our mutation rate is so high, these things are sitting at mutation selection equilibrium. So each one itself is relatively rare, but there's lots of them. And
mhm,
it turns out that somewhere, the best estimates at the moment are somewhere between 5 and 10% of the human population has a rare genetic disease. I mean, it is a strikingly high number. And I sort of tried to do a sort of similar calculation for cod, for example, because we know cod's mutation rate, et cetera, et cetera. And it would be about an order of magnitude lower than that would have a
yeah,
rare genetic disease in
mhm,
cod. Not that we would know, because cod never turn up to the vets to get inspected. I mean, in some ways, it's a sort of funny old thing because, genetic disease is also defined in no small part by the fact that you turn up to the doctors. There's almost certainly in that regard, a large numbers of other very rare conditions, but that we do have no name for because they kill you always in utero.
mhm,
So if you had a really essential gene, for example, that if this gene is gone or mutated, you cannot develop into an embryo, we would never call that a rare genetic disease because you just wouldn't know about it. It would just be yet another pregnancy that was vanished, just a
right,
ghost pregnancy. It may have started, but it didn't get anywhere, or may just simply not start. In which case, that whole bunch never get classified. So the mutations that actually cause rare genetic diseases are actually a subfraction of all the harmful mutations. The very most harmful we never get to see because they kill you in utero.
yeah,
And so they don't get a name and they don't get classified because they kill you in utero.
right. So can you talk a little bit about, as I said before, I heard you say that medicine is
Yeah.
anti-selection.
Yeah, so this is very much my opinion of things. So if you have take the understanding that what is selection doing in the case of rare genetic diseases? What selection is doing is go, oh, that's harmful out of the population. That the death of the kid who's got early cancer because of a new mutation, that is simply selection operating to move that mutation out of the population. Now, what does medicine do? Medicine effectively is anti-selection in the sense that it keeps it in the population because we want to save the kid. So typically what we do is we save the kid, but we don't change the gene. So the gene stays in the population, but the kid survives. And ethically, I think that's absolutely the right thing to do. That's what medicine does. So most of the time, what medicine is doing is keeping people alive who should have died. Should not ethically should, but would under any other circumstances if you
Right.
didn't have medicine would have died. So should in an evolutionary sense. Were fated too, but medicine keeps us alive, and that's true also for vaccines, antibiotics, and so on and so forth. So we know, for example, that individuals who die from just about any disease, infectious disease, that is, there tends to be genetical biases. So some people have genes which mean that they are more likely to die of certain infectious diseases if they get them. Some are less likely. And so whenever those infectious diseases come along, you should see the combination of the genetics plus the infectious disease will kill some individuals. Infectious plus genetics, they will be all right. So, for example, for something like sepsis, blood infection, bacterial blood infections, we know that there are certain genetic variants in a gene called caspase. For example, these genetic variants mean that one variant means that you're very likely to die if you get sepsis and much more likely to get sepsis. The other means you can have exactly the same bacterial infection, be unlikely to get sepsis and unlikely to die. But the point about medicine here, the medicine is antibiotics. So we give everybody antibiotics and everybody survives. So we have blunted selection's effect again. So the ones who would have died with this bacterial infection aren't now. And the ones that wouldn't have died but would have been a bit poorly for a bit, they survive too. So everybody survives with antibiotics. Likewise, vaccines, we're having exactly the same effect. So both genetic diseases and infectious diseases, what medicine is doing is keeping people alive. That's the function of medicine. But if you're keeping people alive who genetically otherwise would have died, that's anti-selection. But it's a good thing. That's why we've
Yes.
got big
Yep.
brains. It's a
Mhm.
good thing. Medicine isn't this arbiter going, oh well, Darwin says, your genes aren't very good, you should die, therefore I'm not going to give you medicine. No, no, no. That's abhorrent. That's appalling. And so no, I would say basically the function of medicine is to dampen the effect of natural selection, because most of the time what natural selection does is kick mutations out of populations, those that are harmful. It's making the harmful ones less harmful. So if you had the mutational type that meant you would have gotten sepsis with a bacterial infection, but you now survive, there's a death that's been prevented. If you have childhood cancer and we can now cure those childhood cancers, there's a death that is now being prevented, and so on and so forth.
Right. Yeah. This is just a personal hypothesis that I have. I don't know that it's been studied at all. But when you look at the so-called autism epidemic, first of all, the vast majority of it is just our ability to identify and autism, and also the way it's been categorized. There's been more disabilities categorized under autism spectrum disorder as times went on, which increases the number. But I think a possible explanation for a small portion of it could be what you're talking about, where in the 50s, 60s, 70s, maybe someone who was speech delayed and displaying like stereotypical behaviors would have been institutionalized at a young age, or been at least on the fringes of society. And now, as we've advanced our understanding through medicine, advanced our ability to treat autism, those people are better integrated into society and have a better chance to have a family. And so as those genes get passed on, that might cause a small uptick. This is my theory, by the way. This is not Dr. Hurst's
It's a
theory.
perfectly reasonable hypothesis. I would like to see it tested. But
Mm,
I know little about autism. The colleagues who tell me say, yeah, but a big component of it is we are simply diagnosing more and better. But the other thing I think that's striking about autism that I think is very well worth reiterating here today is that there used to be, I don't know if you're old enough to remember it, but there used to be a big push when a kid had autism to basically blame the parents. Usually the mother, of course, blame the mother. That it was something to do with the mother brought up the kid and they didn't bring the kid up right. And it was
yeah,
blame the mother.
the
And
so-called
I have a horrible feeling that when it comes to genetic diseases, people are doing the same. And please Do not blame yourselves.
Yes.
This is the most awful thing to happen.
Mm,
You see a disease and we don't know the explanation for it. You're suffering enough as it is. This is nothing to do with anything you did wrong. This is simply bad luck. Most cancers are bad luck. If you have a kid with a genetic condition, it is simply bad luck. You did nothing wrong. We're just an unfortunate species with a horribly high mutation rate, and this is happening all the time. You did nothing wrong. And if somebody blames you or you are blaming yourself, and I can imagine how you might turn inwards and blame yourself. Please, please don't. This is just evolution playing out, as it were. We have a high mutation rate because selection is inefficient. The consequence of that is 5 to 10% of a rare genetic disease. Blame yourself for that, and you may as well blame yourself for everything good evolution has ever done,
right. Yes.
which is equally silly thing to do. No, don't blame yourself. So yeah, no. Do you know that going back to your interesting possibility of these individuals being institutionalized once upon a time,
Mm.
there is, the case of Buck versus Bell. So this is going back to 1920s in America. So
That
some
was
American
uh,
states,
forced sterilization,
yeah,
right?
forced sterilization. This the Buck versus Bell case went all the way to the Supreme Court to ask, does a state have a right to forcefully sterilize? It was nearly always women, by the way. And it was just the state sort of deemed you're somehow deficient, and so we're going to sterilize you. And amazingly, the Supreme Court decided 8-1 in favor of the states that they did have a right to forcefully sterilize people. I think forceful sterilization, when that was eventually abolished, then gave way to institutionalization instead.
Right. Yeah.
But I think your logic is sort of very similar. But I don't know. I really don't know enough about autism, with the exception to say it's quite clear autism is a highly genetic condition.
Yeah. One quick point, and then I know we're running up on time, but I don't know if you're familiar with Dr. Temple Grandin at all. She's a professor at Colorado State University.
I fear not.
So she is autistic, and she's older, I think, in her like 70s or so, but she was almost institutionalized as a child.
Wow.
And her mother, because she was like 4 or 5 before she started talking, and her mother ended up finding a speech therapist who was able to work with her. And it turned out that like she just had a hard time processing language.
Mhm.
You had to talk to her really slow so she could process all the words. And once she was able to kind of get rolling, she's become, a professor at a college. She's profoundly impactful on animal science. Like if you get a fast food burger from, or yeah, if you get a burger from a fast food restaurant, the meat packing plant that burger came from is based on designs by Temple Grandin. Like she was
Oh.
revolutionary in animal welfare and sort of um ways to make the whole process much more humane on cattle, among other things. And like if
Interesting.
you see, I don't know if you've ever seen on a farm that they need to like do something to a cow, like give it a medication or an injection or whatever. They put it in what's called a squeeze chute,
Yep,
where
no
they
thing.
walk in and then it kind of clamps down on them and squeezes them, and that pressure kind of helps keep them calm and keep them still while they're working on it. Dr. Grandin invented that originally
Oh,
for herself because she needed that deep pressure as an autistic person,
how interesting.
but she didn't like human contact, so she didn't want hugs. So that's just an interesting aside.
Well,
But
the only thing I'd add to that is, do you know our uber intelligent spy agency in the UK,
mm,
GCHQ? Yeah, these are the ones who process all the encrypted information and all the, yeah, they're basically backroom spies, as it were. Interestingly, they like to recruit neurodivergent and autistic people because they think differently.
sure, absolutely,
And I think when I discovered it, I thought that's really interesting because, as you say, not that long ago, these would be simply dismissed. And I go, no,
right.
no, no, these real value here,
Mm.
and so on and so forth.
Yeah. Alrighty. Well, like I said, I know we're running over on time, so I really appreciate you taking the time to talk to us to share your knowledge with our audience. Again, Dr. Hurst's book, The Evolution of Imperfection: The Science of Why We Aren't and Can't Be Perfect, published by Princeton University Press, is out now. I've read it. This is going to seem like a little bit of a backhanded compliment, but you would expect a book covering evolution to like kind of inevitably be pretty dry. And I was really impressed with how sort of light and engaging you were able to make it when covering such a technical subject.
Well, I'm
I
delighted. I'm
really
delighted to hear that, and yeah, it was intended in no small part for your sort of audience as well. It's important, I think, people know that both they're not alone and they're not to blame. We just have a stupidly high mutation rate, and the consequence of that is there are a number of really unfortunate parents in many regards who have very sick kids, and my heart goes out to them.
I appreciate it. Alrighty, and as always, our listeners can find us on Facebook. If they need to get in contact, they can email us at mef2cast@gmail.com. Thank you, everybody.